Now we move into chemical digestion and absorption. We'll begin with carbohydrates. Starch is the most digestible carbohydrate. Cellulose is another carbohydrate, but it's indigestible for us. However, it is important as a source of fiber. Glycogen is digested in a similar way to starch. However, our consumption of glycogen is negligible. So starch is going to be broken down stepwise. First, into oligosaccharides, which are molecules made up of about 8 glucoses. Then those 8 glucose oligosaccharides will be digested into disaccharide, maltose. And then finally, maltose will be split into glucose, and glucose can be absorbed by the small intestine. Now this is why there's a big difference between eating complex carbohydrates, such as starches, and simple carbohydrates, such as straight glucose or high fructose corn syrup, because starches need to be digested stepwise, whereas glucose or sucrose is available for immediate absorption. So it tends to mess with our blood sugar levels a lot more. The process of carbohydrate digestion, as we've already learned, begins in the mouth with salivary amylase. Salivary amylase is going to hydrolyze starch into the oligosaccharides, and it works best at the pH that exists in the oral cavity. Once amylase reaches the stomach, it's quickly denatured, and then it's digested by pepsin. It's treated just like any other dietary protein. Salivary amylase digests about 50% of the dietary starch before it reaches the intestine. Once the starch reaches the small intestine, it's all up to pancreatic amylase. Pancreatic amylase is able to convert the remaining starch into oligosaccharides and maltose, and it does that within about 10 minutes. The oligosaccharides and maltose can then contact the brush border enzymes, which you'll remember are stuck in the brush border on the microvilli. The enzymes are dextranase, glucoamylase, maltase, sucrase, and lactase, and they each act on a specific sugar to turn it into glucose. Lactose is an interesting sugar because for us humans, it becomes indigestible at age 4. However, many of us continue to drink milk. We'll look at that in just a moment. Now on to absorption. We've got the carbohydrates broken down into the simplest monomers possible. The plasma membrane of the absorptive cells, so the simple cuboidal cells, has transport proteins that absorb monosaccharides. So here we are in the lumen of the small intestine. We have transport proteins that are specific to absorbing each of the variety of monosaccharides. 80% of our absorbed sugar is actually in the form of glucose, and it's taken up by sodium glucose transport proteins at the microvilli from the inside of the small intestine. It's transported across, and then it moves by facilitated diffusion out into the extracellular fluid of the villus. This increases the osmolarity of the fluid in the villus, and thus it draws water osmotically from the lumen of the small intestine between the cells and into the villus. With water comes more glucose and other nutrients by solvent drag. Now the tight junctions naturally are leaky between epithelial cells. The sodium glucose transporter can absorb glucose, galactose, and fructose, again absorbed by the capillaries in the villus, and then the hepatic portal system delivers all of these nutrients to the liver for further processing. Lactose intolerance then is kind of an interesting issue because after the age of 4, most humans don't have the enzyme lactase to break down the sugar lactose. And so it passes undigested into the large intestine and wreaks havoc on many people's digestive systems. First of all, it increases the osmolarity of the intestinal contents, which causes water retention in the colon and thus diarrhea. Also, bacteria will break down the lactose by fermentation, which results in the production of gas. So lactose intolerance has quite a variance in the way it displays itself. Some people simply have gas, and other people have very strong, painful contractions in their intestinal tract. It occurs in many parts of the population, pretty much anywhere that hasn't had a history of milking domestic animals. This practice goes back about 10,000 years. In humans, we're the only animals to milk other animals and consume their milk. People who are lactose intolerant can consume yogurt and cheese since these have been cultured with bacteria and the bacteria break down the lactose. However, straight milk, not so good. So as you know, proteins are made up of amino acids. The amino acids that are absorbed by the small intestine come from three sources. First, there are dietary proteins. Those are the ones that we consume. Second, there are digestive enzymes that have been digested by other digestive enzymes when they're worn out. And then sloughed epithelial cells. As the epithelial cells make their way up from the intestinal crypt to the tip of the villi, they're sloughed off. And those will be digested by enzymes. So our endogenous amino acid consumption, as in the ones that we break down from within our body, makes up about 30 grams per day. Our exogenous amino acid consumption, that we actually bring into our body from our diet, is about 45 to 60 grams per day. Enzymes that digest proteins as a general class are called proteases or peptidases. Pepsin is the one that we'll see working in the stomach. Pepsin works best at a pH of 1.5 to 3.5, which is perfect in the stomach. So pepsin will digest about 10 to 15% of dietary protein into shorter peptides and some free amino acids. Peptin hydrolyzes bonds between tyrosine and phenylalanine. Any bond between tyrosine and phenylalanine, pepsin is going to chop, and thus we end up with the dietary proteins being cut into shorter pieces. Once pepsin moves from the stomach into the small intestine, of course, the pH becomes more alkaline, and thus it no longer functions. So when we get into the small intestine, trypsin and chymotrypsin take over the process of digesting proteins. As you'll remember, trypsin and chymotrypsin are products of the pancreas. They'll hydrolyze peptides into even shorter oligopeptides. And then those oligopeptides will be taken apart one amino acid at a time by three other peptidases. There's carboxypeptidase, aminopeptidase, and dipeptidase. Now, carboxypeptidase is going to remove amino acids from the carboxyl end of any polypeptide chain. Aminopeptidases chop off amino acids from the amino terminal of any amino acid, and then dipeptidases will split dipeptides right down the middle to release two free amino acids. Carboxypeptidase, you'll remember, is a pancreatic secretion. However, aminopeptidase and dipeptidase are both brush border enzymes. In this figure, we can see the digestion of proteins by pancreatic enzymes, trypsin and chymotrypsin, and then by carboxypeptidase. Once they're broken into smaller subunits, our brush border enzymes can finish the task, producing some free amino acids that then are absorbed into the intestinal epithelial cells. They're also transported by sodium-dependent amino acid cotransporters in a similar way that monosaccharides were transported across the epithelial cells. And then again, facilitated diffusion moves the amino acids out into the bloodstream on the other side. An interesting fact here is that infants absorb proteins by pinocytosis, and this allows them to obtain the maternal immunoglobulin A so that they can get some transferred immune benefits from mother's milk. And this is released directly into the blood by exocytosis. This is very beneficial to the infant. However, it does have its disadvantages. The disadvantage is that intact proteins entering the infant's blood can sometimes be detected as foreign antigens and thus could trigger food allergies. As the intestines mature, their ability to perform pinocytosis declines, but it never completely ceases. And this may explain adult onset food allergies. Now pause the recording here for a moment and draw an intestinal epithelial cell. Diagram the digestion and absorption of starch and proteins. Now we move on to lipid digestion and absorption. Lipids are hydrophobic, which makes their digestion and absorption a little more complicated than carbohydrates and proteins. Fat digesting enzymes are called lipases in general. We've already seen lingual lipases, which were secreted by the intrinsic salivary glands of the tongue. And they're active in the mouth and become even more activated once they reach the stomach and work in conjunction with gastric lipase. Here they're able to digest about 10 to 15% of lipids before they even reach the duodenum. Once we do get to the duodenum, pancreatic lipase is going to digest most of the fats. Fats enter the duodenum as large globules, which isn't very functional because they can only be exposed to lipases at their surface. So the globules need to be broken up into smaller emulsification droplets by certain components of the bile, which are lecithin and bile acids. It's the agitation of segmentation that breaks up the fat into these droplets, and the coating of lecithin and bile acids that keep it broken up. This exposes far more of its surface area to the enzymatic action. There's enough pancreatic lipase in the small intestine after a meal to digest an average daily fat intake in as little as 1 to 2 minutes. These lipases act on triglycerides. They remove the first and third fatty acids from a glycerol, which leaves us with 2 free fatty acids and 1 monoglyceride. Now, how do these free fatty acids and monoglycerides become absorbed? This depends on some minute droplets in the bile called micelles. They're made in the liver. They consist of 20 to 40 bile acid molecules, and they aggregate with their hydrophilic side groups facing out and their hydrophobic steroid rings towards the inside, so they can easily float around in the aqueous environment of the chyme. Bile, phospholipids, and cholesterol will then diffuse into the center of the micelle to form its core. Then the micelles pass down the bile duct into the duodenum. In the duodenum, they're able to absorb these fats, including fat-soluble vitamins, cholesterol, all of the free fatty acids and monoglycerides that are produced by fat digestion. They're going to transport these lipids to the surface of the intestinal absorptive cells. Here the lipids will diffuse out of the micelles and in through the plasma membrane inside the epithelial cells. Micelles are then reused. They travel away from the brush border and pick up other cargoes of lipids and transport them to the absorptive cells. Once we have the monoglycerides and free fatty acids inside the epithelial cells of the intestinal wall, they're transported to the smooth ER. The smoothie art then resynthesizes them into triglycerides, which the Golgi complex coats with phospholipids and protein to form chylomicrons. These chylomicrons are packaged into secretory vesicles that will migrate to the basal surface of the cell, release their contents into the core of the villus, which is where they're taken up by the porous lacteals. Once this fatty intestinal lymph, which has this sort of white color, it's called chyle. Here we can see the chylomicrons in secretory vesicles being dropped out into the extracellular fluid and then absorbed into the lymph. So now take a moment to diagram fat digestion and absorption. Consider the emulsification process, the hydrolysis of fat into monoglycerides and free fatty acids, its transport in my cells to the brush border, and then its transport within the cell and out from the cell in chylomicrons towards the lymphatic vessels. So the nucleic acids, DNA and RNA, are present in much smaller quantities than any of the polymers discussed previously. First of all, they'll be broken down by nucleases, which hydrolyze the DNA and RNA into their monomer nucleotides. Then nucleosidases and phosphatases that are located in the brush border split them further into their base components of phosphate ions, either ribose or deoxyribose sugar, and then nitrogenous bases. These products are then transported across the intestinal epithelium by membrane carriers, and they enter the capillary blood of the villus. Vitamins are absorbed unchanged. There are fat-soluble vitamins, such as vitamin A, D, E, and K. They have to be absorbed with other lipids. So if someone's consuming a diet that is void of lipids, then fat-soluble vitamins cannot be absorbed, and they'll just end up being in the feces and wasted. Water-soluble vitamins, however, the B complexes and C, can be absorbed by simple diffusion. However, as we've already learned, B12 needs to be bound to intrinsic factor, and that's because it's so large. Once it's bound to intrinsic factor, it can be endocytosed by cells of the distal ileum. So finally, we'll take a look at the absorption of minerals and water. Minerals are the electrolytes, and they're absorbed all the way along the small intestine. Sodium, as you recognized, was co-transported with both sugars and amino acids across the brush border of the intestinal cells. Chloride was exchanged for bicarbonate, reversing the chloride bicarbonate exchange that occurs in the stomach. Potassium is absorbed by simple diffusion. As the concentration of the chyme rises, water is absorbed, which creates a gradient that's favorable for potassium absorption. Iron and calcium are a little bit different in that they're absorbed in proportion to the body's needs. Most other minerals are absorbed at fairly constant rates, regardless of needs, leaving it to the kidneys to excrete any excess. However, iron and calcium absorption are under fairly strict hormonal control, and thus only the amount needed is absorbed. So the digestive system is one of several systems involved in water balance. It receives about 9 liters of water per day. Most of this is from gastrointestinal secretions. It comes in the bile, comes in the pancreatic juice, comes in secretions into the stomach and other secretions into the small intestine. That's about 6.7 liters a day. We drink another 1.5 liters or more each day, and then we'll have about 0.5 liter to 1 liter in the food that we consume each day. Now, 8 liters of that is absorbed by the small intestine as water travels by osmosis down its gradient into the tissues. About 0.8 liters is absorbed by the large intestine, and then only 0.2 liters is left over, and that's voided in feces each day. Diarrhea results when the large intestine absorbs too little water, leaving it in the feces. This happens when feces passes too quickly. If the intestine is irritated, maybe from some contaminated food, or if the feces contains high concentrations of solute, for example, lactose or other sugars, which is going to draw water from the tissues into the intestine. When there's not enough water in the large intestine, too much water has been reabsorbed, that results in constipation. The feces have become hardened and they don't move as fluently through the large intestine. Now, this completes our unit on digestion and absorption. You have a few diagrams you've completed, and we'll take a look at those when we meet in class. So now we'll move on to look at the final section of this chapter, the large intestine.